My name is Phoebe Jekielek. I am a PhD candidate at the University of Maine in the School of Ecology and Environmental Sciences, and I'm working with Maine-eDNA. I am evaluating environmental DNA as a tool for monitoring commercially important fisheries, both from an aquaculture perspective and a wild harvest perspective. One of the things I'm trying to do is I'm trying to relate quantities of different life stages of scallops, for example, so larvae or gametes or adults, to gene copy number in the water. That is something that I need to do before I can really start to interpret and think about other applications for eDNA in monitoring shellfish populations. One of the things I'm evaluating is the spatial and temporal variation in scallop eDNA signals above a wild harvest bed. So I have a population of scallops, I know that they're there, and I take samples at different depths to look at the spatial variation in that scallop signal from bottom to the surface of the water. And I monitor that over a long period of time to see how that signal changes over multiple months. So I think one of the things that we're really trying to tackle is how can we use environmental DNA as a tool? And for any given species, there are a lot of different questions to answer if you want to use it for a species. So for something like scallops, that isn't a commercially important species, that really is quite sessile, so it doesn't move around a lot, it kind of just sits on the bottom, it might be a great tool. So can we just take a water sample and say there's this many scallops on the bottom? Maybe not, but we might be able to take a water sample and understand how many larvae are in that water sample, or if there are scallops present or not. One of the things that I've really, I think, focused on during my process is thinking about all of the different angles that I need to come at attacking this question. I don't know if an egg gives a different DNA signal or different DNA quantity than a larvae, or an adult just being an adult scallop sitting in the water. So really kind of figuring out how all of these measurements interact and then how we can apply them in the field to answer questions that are commercially important is really what I'm interested in. I think eDNA can be a helpful tool for answering some of these questions because all you do need is a water sample. You might not need to go out and drag up a bunch of scallops, or you might not need to wait to put out a spat bag and then wait 5 months to retrieve it to see how much larval supply is coming through an area. So I do think thinking about how accessible eDNA can make sampling is really important. And I think that in this whole program, we're really teasing out a lot of those questions for different species and different applications. I think it has a lot of potential for helping us to understand, especially in known species that are challenging to sample, like scallops that live on the bottom of the ocean, where you either need to dredge them up or go diving. It has a lot of potential in helping us gain more access to understanding their cycles, understanding how they interact with the environment around them, and what it might mean for commercially important industries like aquaculture or wild harvest.